Okay, so now we move on and we start very gradually making a transition to molecular genetics. That's where we're going. So you are now familiar with what is a gene. It's basically a segment of DNA that has a region that will code for a protein. But that's not all that is part of the gene, correct? You also need to have a ribosome binding site. You need to have a region that starts up transcription. You have to have a promoter, and in eukaryotes you will also have enhancers and other sequences, splicing, etc., etc. So when all of those rules are present on a segment of DNA, that DNA will produce a protein, and that protein will produce a phenotype. That wasn't known in the 1930s, 1940s. There were a series of experiments that were done by Beadle and Tate that basically gave us this picture of what is a gene. And so the original hypothesis that they came up with was that one gene codes for one enzyme. Since then, we've modified it to be one gene, one protein. And of course, there are some examples where it's just one gene, one RNA. Those are the ones that don't make proteins. The RNAs, the RNAs, like transfer RNAs and ribonucleotides. But on the average, in humans, there are about 25 to 30,000 protein coding genes. Each gene makes one protein. Again, that rule is not fixed in stone, but simplicity's sake, we'll say one gene, one protein. And the original hypothesis. So we'll look at the experiments that were done to get to this. Okay, so in the next few lectures, we're going to answer the next few questions addressed to what is the structure of a gene? And you've already been exposed to that in the very first lecture on transcription and translation. How does a gene function? And how is the information stored on the gene? So we're going to go back in a sense to chapter 8 to some extent. Okay. And the relationship between genotype and phenotype. Okay, so biochemists who've been working on specific pathways have been working on these pathways for many, many years. And they knew, for example, the pathway that generated arginine. You start with glutamic acid, which is down here. Glutamic acid is converted to ornithine. Ornithine is converted to citrulline. Citrulline is finally converted into arginine. So we knew the intermediates in the pathway to the production of arginine. Beadle and Tatum wanted to understand how arginine is made in a cell. Not biochemically in a test tube, but within the cell. Now, within cells, there are protein molecules, enzymes, that will take a substrate and convert it into a product. You know the mechanism, it lowers the activation energy, allows the chemical reaction to proceed. So that's the function of an enzyme without actually being part of the product being made. They have autonomous entities. So how are these enzymes affecting this pathway, and how are these enzymes made in the cell? So how do you link the DNA sequence to the protein or the enzyme that carries out this part? And that was what Beadle and Tatum were focused on. They were focused on figuring out the genes that were involved in the production of arginine in specific cells. And they were working with neurospora, which is bread mold. So the experiments of Beadle and Tatum in the 40s provided the first insight into gene function. And they developed the hypothesis that one gene codes for one enzyme molecule, one type of enzyme molecule. And the hypothesis had three specific points. The first, products are synthesized as a series of steps, and that was known from the biochemistry. So you had a specific product, and there were intermediate steps, and then there was a substrate. So from substrate to intermediate to product. Each step is is catalyzed by a unique enzyme. So step 1 to step 2 is catalyzed by one enzyme. Step 2 to step 3 is catalyzed by completely different enzyme. That was the second point. And then the third point, which is the most important one for this class, each enzyme is specified by a unique gene. So there is a relationship that builds up in this part. Imagine you have a precursor, mixed intermediate 1, mixed intermediate 2, and then the product. That's the first part. Products are synthesized as a series of steps. You go from precursor to intermediate 1 to intermediate 2, finally to a product. The second point is each step in this conversion process is categorized by a unique enzyme. So you have enzyme A that takes this precursor, converts it into intermediate 1. Then enzyme B takes intermediate 1 and converts it into intermediate 2. And then enzyme C takes intermediate 2 and converts it into a product. And I just remembered I had to make an announcement. Somebody just informed me that if you want to withdraw this class, the last date for withdrawal is on Friday this week. Okay? So, not that I want you to withdraw, but I'm just kind of giving you this. I have to make that announcement. I'm sorry, I just got to do this. Anyway, so you have 3 points. Products are synthesized in a series of steps, and each step is catalyzed by a unique enzyme. And then, each enzyme is specified by a unique gene. So there is gene A on the chromosome that will produce enzyme A, and it's enzyme A that will take precursor and convert it into intermediate 1. There is gene B that will make RNA. The RNA will be translated into a protein. The protein will be enzyme B. This enzyme B will take intermediate 1 and intermediate 2. And gene C will make enzyme C, which will convert intermediate 2 to product. So what happens if you have a mutation in these enzymes? A mutation is something that disrupts the enzyme function. So if you have a mutation in gene A, would you make product? Yes or no? No. The reason, if you have a mutation, this enzyme would not be made. If this enzyme is not be made, this precursor cannot be converted into intermediate 1. If you can't convert this into intermediate 1, you're not going to be able to make a product. So, mutation in gene A, no product. Mutation in gene B, also no product. Mutation in gene C, also no product. So, what are the consequences of a mutation in gene B? Let's say we have a mutation here. We're blocking the step over here. Would we make a product? No. Would we make intermediate 2? No. What about intermediate 1? Yeah. Because the roadblock along this pathway is right here. Because the roadblock is here, you would make intermediate 1, but then you couldn't convert it to intermediate 2. Now, enzyme C is present in the cell. Okay? But enzyme C cannot make the product because there is no intermediate 2 in the cell. So the pathway is blocked, and so you just can't carry on. When you have a mutation in gene B, you're basically stopping the pathway. The presence or absence of gene C is immaterial. It's almost as good as not having this enzyme being made. Would the final product be produced? The answer is no. Would intermediate 2 be made? Would intermediate 1 be made? What happens if we add intermediate 1 to 2? So here's the next question that we're going to look at. So we have a mutation here. We have a roadblock over here. Now let's imagine you could feed this cell so that it cannot make this product. Let's imagine you can feed the cell this intermediate. Would it be able to make product? No. Okay, we have a roadblock here. We're going to feed the cell intermediate 2. Will it now be able to make product? Yes. Because now you're giving it intermediate 2. Now the cell can convert it into a product because gene C is still present. So even though you have a roadblock, you can still go ahead because you've artificially fed it intermediate. And this is basically the principle that Beadle and Tatum used in their experiments. And we'll go through those experiments. So here's Neurospora, a colony of Neurospora. It's a haploid fungus. It's the red mold that you observe on old bread. And there was an important aspect to this experiment that allowed them to perform this. It could be grown on defined growth medium, i.e., if you gave them very specific chemicals, the cells would grow. If you gave them particular salts, like sodium citrate, potassium phosphate, and ammonium nitrate, and magnesium sulfate, you don't have to know all these. But you gave them certain salts and certain sugars. That's all you have to give them: salt, sugar, and water. You give them these three things, the cells will be able to grow. You don't have to give them amino acids, you don't have to give them carbohydrates, well, except sugars. You don't have to give them fats, you don't have to give them any of these other acids. They will make everything you need from these few ingredients that you're providing them. Basically, sucrose and these salts. Nitrogen source, oxygen source, hydrogen source. Okay. Neurospora can then synthesize the amino acids, the vitamins, and everything else that they are required to grow and form a colony on a plant. So, salt plus sugar, and voila, you get growth. Okay, so a few terms. A prototroph is a strain that utilizes sugar, salt, and water to grow. You could call it a wild strain. Biologists call them prototrophs. An auxotroph is a mutant strain that cannot grow on sugar and salt and water. It needs additional elements. So, auxotroph is just another word for mutant. Prototroph is just another word for wild. Okay, it may need a specific amino acid. Okay, so here's the arginine biosynthetic pathway. Okay, so the way the experiment was done, I showed you the pathway: ornithine, citrulline, and arginine. Three steps of the pathway. You start with the precursor, which is usually glutamic acid. And what Beadle and Tatum basically decided was they were going to take Neurospora. It has about 15,000 genes, and only a few handful of genes are required for making arginine. So in this collection of 15,000 genes, they had to find a few genes, 3 or 4 genes, that when mutated would stop the cells from making arginine. That was the challenge in the 1940s. So keep in mind, in the 1940s, we did not even know the structure of DNA. Right? So before the DNA had been discovered. So how do you identify 5 genes out of 15,000 genes? And this is a method that is used even today. If you are studying a complex pathway, whether it's aging or diabetes or Alzheimer's or whatever, in theory, this is the pathway method you use to figure out the genes involved in that complex phenotype. Start with a cell or a strain that is normal. And you just at random mutagenize it. Just kind of blast it with mutagens, either chemical mutagens or physical mutagens like radiation. For example, if you were to do this experiment with Neurospora, you would take Neurospora, you would put it in a vial, you would put it into an X-ray source and bombard it with X-rays. At random, the X-rays would mutagenize the DNA. You would make breaks in the DNA. And because you had a million or 10 million cells in that little vial, one cell might get mutated on gene A, another cell will get mutated on gene B, and so on. Once you have those mutants, then you have to sit and sort them out and figure out which mutant is mutated for the pathway you're studying. And that's the exact same method we used. It's the method that was used that got people Nobel Prizes for figuring out the cell cycle. It was the method that was used that got them Nobel Prizes for figuring out development of the software. Same method, same approach. Start with the normal cell, bombard it with mutagens, and then sort out the mutagens and study each mutant. It is known as the awesome power of genetics. You can't do that in biochemistry as easily. So, typically, an organism is taken and it is exposed to a strong mutagen. And this will randomly mutagenize genes. So, they're just cut a shot. In effect, if I had a shotgun and I went boom, some of you would get hit, others would not get hit. It's known as shotgun mutagenesis. I'm not being crazy here, don't go start calling 911. But it's known as shotgun mutagenesis because it's a random approach. There's no specific bullet for each cell. You just hit them at random. Half the cells might not even get mutagenous, they'll be normal. Only some cells will get different mutagens. Once you mutagenize those cells, then you look for the mutant in the pathway of its use. And it's tricky, and it's difficult, and it's doable. So that's why I want to take you through this, because this approach is still used even today in many instances with complex genomes. Okay, so arginine biosynthesis pathway. You take cells or spores and you irradiate them to mutagenize them. Then you take those spores and you grow them on medium containing arginine. You want to study arginine. You've mutagenized the cells. And they are all stressed out, each individual cell, right? Because they've been hit with mutagen. So you give them a nice food source so that they start to grow. So you grow them in the presence of arginine initially. First step, you let them grow in arginine, even though you want to study arginine problem. And then, after they've grown in arginine and formed a nice colony, you transfer them to media lacking arginine, and you ask, can those cells make arginine or not make arginine? So you first grow them, let them grow up after mutagenesis, and then you ask those questions, can you grow in the absence of arginine? And the ones that can't grow in the absence of arginine, the mutant cells that can't grow in the absence of arginine, are the ones that are mutagenized for that part. So, do they grow or do they not grow? If the cells cannot grow on medium lacking arginine, then they must have a mutation in the gene required to make arginine. All right? Is that clear? So, and so then you can say, okay, we have now got a mutant in the gene required to produce an enzyme that is required to make arginine. So you go through yourself that. So, here are the cells. You irradiate them with X-rays or gamma rays or chemicals. Take the mutant spores, you place individual spores on complete media. Complete media is basically media that contains sugar, salt, water, but it also contains vitamins, amino acids, and other ingredients. So you give the cell everything, so it does not have to make anything. Even if it is wild type or mutant, it grows happy. So this is known as complete media. So initially, that's what you do. You grow them up. So you take individual cells, you plate them on a plate with complete media. So this cell will form a colony on the plate and grow. This cell will form a different colony and grow. This cell will form a different colony and grow. This cell will form a different colony and grow. And you get in the picture what happens. So all of these cells get plated on complete media after they've been mutagenized. And they all grow and form colonies. Because it doesn't matter if they have mutations or not, you're giving them everything. And so they're able to grow. Right? This is again another horrible analogy. Right now, all of you can climb trees, okay? Coconut trees. Right? Shimmy up the tree and you can grab coconuts and you can eat them. Right now, all of you can do that. Let's imagine I walk around with a hockey stick and break some of your arms. Now now, some of you are unable to climb up those coconut trees and grab coconuts, so you can't live. If you had to live only on coconuts. But if I were to give you food, you would be okay. You could survive. So that's what this is. As long as I keep giving you food, you're surviving. But the minute now I withdraw the food and say, okay, guys, climb up and get food, there's going to be a group of you who are not going to make it. That's the equivalent. Okay? So you've got the cells, they're all growing because you've provided them with all the ingredients. Then you transfer them to minimal media. You transfer them to media that can take only water, sugar, and salt. No vitamins, no amino acids. Okay? And you ask the question, which of these cells can grow now under these minimal conditions, and which of them can't grow under these minimal conditions? And you might find something that looks like this. These cells can still grow on minimal media, but these two cells are unable to grow on minimal media. Number one, when it's transferred to minimal media, stops growing. And number 7, when it's transferred to minimal media, stops growing. So you know that cell 1 and cell 7 have a mutation, and because of this mutation, they either can't make vitamins or amino acids. Because the thing you've withdrawn from this mixture are vitamins and amino acids. Everything else is still the same. You've withdrawn vitamins and amino acids, so those are the 2 ingredients that are missing. So you can say these cells have mutations such that they cannot make either vitamins or they cannot make amino acids. Right? Okay, so strains 1 and 7 can grow on complete media, but not on minimal media. They have a mutation in the gene required for growth on minimal media. So they have a mutation in the gene required to synthesize either vitamins or amino acids or both. That's the conclusion we need. Okay, so that's basically that. Okay, so now we have 2 strains, 1 and 7. Effective in either amino acid production or vitamin production. Now we take strain 1 and focus on strain 1 only for now. We grow it on complete media, of course it grows. Right? Take strain 1, transfer it to complete media. Next we're going to transfer it to minimal media, just salt and sugar, and of course the strain can't grow. We already did that experiment with that. Here is minimal media with amino acids. So you give them amino acids but not vitamins. And ask, does strain 1 grow? And the answer is, it doesn't grow. Then you transfer it to minimal media with vitamins but no amino acids. And you ask, does the strain grow or not grow? And the strain grows. So what's the conclusion? The conclusion is strain 1 is defective in the production of vitamins. Because when you feed it vitamins, it is able to grow. When you feed it amino acids, it's not able to. Right? So strain 1 has a mutation in a pathway required to produce vitamins, not in a pathway required to make amino acids. Next, you focus on strain 7. So initially, we've come to the conclusion that strain 1 and 7 were defective in either amino acid production or vitamin production. We know strain 1 is defective in vitamin production. What about strain 7? You do the same experiment. You start with complete media, strain 7 grows. You transfer to minimal media with amino acids, strain 7 grows. Minimal media on its own, strain 7 doesn't grow. Minimal media with vitamins, strain 7 doesn't grow. And so based on this, you will say strain 7 requires amino acids. It's got a mutation in the pathway for the synthesis of amino acids. But when you provide them with amino acid, the strain can grow. It's basically able to bypass the requirement because even though it's mutant for this pathway, when you provide the amino acids, And so the conclusion is that strain 7 is defective in the production of amino acid, and the mutant is rescued by addition adding back of this amino acid. So now we've added all 20 amino acids. There are 20 amino acids that are the most common amino acids, and we've been adding back all 20 amino acids. We haven't focused on arginine yet. We are on amino acids. So then the next question is, which of the 20 amino acids is strain 7 defective in? Is it arginine, leucine, lysine, glycine, whatever? Right? Out of the 20. So, which amino acids? So when you start with the experiment, you do the same thing. You take minimal media plus vitamins plus all 20 amino acids, you get growth. You get minimal media plus vitamins plus lysine, no growth. Minimal media plus vitamins plus glutamine, no growth. Minimal media plus vitamin plus arginine. That will tell you that strain 7 is a mutation in the pathway required for the production of arginine. And you work your way through all 20 amino acids, and hopefully at the end you will have strains that are only unable to make arginine but can make all of the other amino acids. And you focus on those mutants. Because those are mutants involved in the biosynthesis of arginine. So, when you've done this kind of an analysis, you will find mutant cells that are unable to produce arginine. And Beadle and Dayton identified mutants, which they called argines, ARG. ARG1, ARG2, ARG3. So they identified these mutants, they could not make arginine. That's all they were able to say at this point in the experiment. They had mutant cells with mutations in specific genes that could not make the enzyme, that could not synthesize arginine. Now you know the pathway for arginine synthesis. That was known before Beadle and Tatum set out their experiments. So they knew what the intermediates were in this pathway. Our chemical pathway for arginine synthesis was already known based on work by biochemists. Ornithine and citrulline, which are related to arginine, were the precursors in this pathway. And so the pathway was basically precursor, which is basically glutamic acid, ornithine, citrulline, and arginine. So there's your pathway. And so these were mutants that could not make arginine. So where are these mutants? Maybe ARG1 is an enzyme that converts precursor to ornithine. We'll ignore that step. Let's focus on this step here. Well, you could also do this if you want. I've done this whole pathway. So you have a precursor, converts to ornithine, converts to citrulline, converts to arginine. So there are 3 steps in the pathway, and there are 3 enzymes that are required for this pathway. So the question is arginine 1 going to convert precursor to ornithine, or is arginine 1 going to convert ornithine to citrulline, or is arginine 1 going to convert citrulline to arginine? That's the next question you have to ask yourself when you're doing this experiment. And the same thing with arginine 2 and arginine 3. We know there are enzymes, enzyme 1, enzyme 2, enzyme 3, but we don't know whether these mutations, arginine 1, is a mutation in enzyme 1 or enzyme 2. And so you do the same experiment that you've been doing before, but instead of feeding them arginine, you feed them the precursors. So you start with the minimal media, the salt and the sugar, and now you feed the cells ornithine, or citrulline, or arginine. You feed them just one of these intermediates in the pathway, and you ask the question, what's going on? So imagine there is your actual pathway. Precursor, ornithine, citrulline, arginine, enzyme 1, enzyme 2, enzyme 3. Enzyme 1 is being coded by arginine 2. Enzyme 2 is being coded by arginine 1, and enzyme 3 is being coded by arginine 3. Let's imagine that's it. I've changed this figure from what I had on my slides yesterday. Today morning I changed them, so you guys will have a slight change in your pictures on your slides if you printed them out. Just to make a specific point. So here's mutant 1. It's a mutation called ARG1. But ARG1 is here, which makes enzyme 2. So if I were to, you have a roadblock over here in this mutant. You're not making, there's a mutant R1, you have a mutant enzyme. Because of this mutant enzyme, you can't convert ornithine to citrulline. If I take this cell and add ornithine, will it be able to grow? The roadblock is here. I'm feeding it ornithine. Will it be able to convert it to citrulline? No.